Hello, everyone, and welcome to what has to be, I think this is week 5 or 6 of our journey together in our biology journey studying. So we're going to be looking at some questions, and I would, if you have any questions that you would like for me to look at, you know, feel free, please feel free to ask. But we have some questions that we have to look at that are very important to us. We're going to break it down step by step so that you could understand it. And so let's begin. So the first question that I want us to look at would be about basically about immunity. So can someone tell me, and you could open your mic, can someone tell me what is the function of, in your own words, what is the function of the immune system? When you think about the immune system, what is the function of the immune system? Anyone can answer? Don't be shy. Let me ask. Jasmine, Jasmine, are you there? Where's Jasmine? Check the chat.
I am here
What would you say is the function of the immune system?
to defend against pathogens.
Very good. To defend against pathogens. Very good. Angelina said to protect the body against pathogens, basically saying the same thing. Um, Liam, what what we call the cells that whose primary role is to aid in immunity? What we call those cells?
Will that just not be white blood cells?
The white blood cells. Very good. Um, so good. So now, do you know the cells that are responsible for phagocytosis? Does anyone know those it's the white blood cells, but they're responsible for phagocytosis. Um, can anyone tell me that? What do you call those cells? Anyone? Or maybe I could ask this question if y'all don't know. What is phagocytosis in your own words? So maybe Maria, you could help us with that. What do we mean by phagocytosis? Maria Francis. Oh, can I have a Maria? How about Shekinah? Shekinah, where are you? What do we mean by phagocytosis? Let me see. No, I can't really hear from them. Maybe they just wanna type. So Jasmine, what do we mean by phagocytosis?
Uh, the process by which bacteria is engulfed by
Mhm.
leukocytes to destroy them.
Okay. Okay. So it's basically the engulfing of bacteria. Very good. That's phagocytosis. So so phagocytosis happens in steps. If we were to describe the steps of phagocytosis, can someone outline to me? It don't have to be all the steps. If you could do all the steps, you could give me all the steps. But if you could just give me 1 or 2 steps in the process of phagocytosis. So we know that phagocytosis is when white blood cells engulf bacteria. They engulf foreign invaders, right? They engulf the pathogens, they digest them. So, okay, that's the functions of that is what phagocytosis is. But what are the steps in phagocytosis? Because it happens in steps. Anyone want to try? What are the steps of phagocytosis? You don't have to be in order, just tell me what you know. So that'll be literally our first question, right? So our first question is we want to describe the process of phagocytosis, right? So Sierra, you want to help me?
For phagocytosis, so when the pathogen is engulfed by the cell membrane, um, it is then taken in and the enzymes in the lysosome will
Mm-hmm.
then start to break down and neutralize the bacteria.
Okay.
And then when it's finished neutralized by the lysosome, then all the excess parts are then um exited via exocytosis.
Okay. Sierra, what do we call so here we're talking about phagocytosis, right? In this question, what do we call the white blood cells that are specifically responsible for that?
The neutrophil.
The neutrophils. Very good. Liam, you want to help her in describing phagocytosis, like the steps?
I think my understanding was similar to hers, except I think I would have worded it differently. Like,
Okay, just give us something.
I mean, it has obviously the neutrophils would have to identify the pathogens
Mm-hmm.
and then through endocytosis engulf it. And then
Mm-hmm.
at that point,
Mm-hmm.
um, they would be, what's that bit again? No, say
I
it and I forget it.
I know you're good. You're good. I like that you mentioned the identification part, right? Okay, so
Mhm.
basically, the way I'll describe it, I'll just describe it in just 6 steps, right? So the first step that I'll talk about um this is steps for phagocytosis. So the first step is obviously, if you look at the diagram that they gave us, the first step is definitely going to be attraction. All right. So I'm just going to list the steps. I'm not going to like write all it down, but I'm just going to list the steps. So the first step is attraction. They call that um, chemotaxis. I don't know if you're all familiar with that term, but it's basically attraction. So the neutrophil is going to be attracted to the site of an infection. And so you may be like, well, how is it attracted? Well, the bacteria releases chemicals or damaged tissues releases chemicals that act as like signals, and the neutrophils are gonna be able to understand those signals and go to that area where the bacteria is or where the damaged tissue is. Does that make sense to y'all? So the first step is going to be attraction. So the neutrophil is going to move toward that place where there's like a higher concentration of these chemicals. And again, these chemicals are released by the damaged tissues or they're released by the bacteria itself, and they act as signals to the neutrophils, telling them that, hey, listen, you need to go there because something is happening on. It's almost like a 911 call. That's what it basically is, right? So chemotaxis or attraction is basically you have a movement toward a signal, the movement toward a chemical signal. Hopefully that first step makes sense to you, right? The next step is recognition. So it's going to be recognized. So I think, or some people call it identification. That's what Liam was hinting at. So recognition, right? Recognition and attachment. All right. So that's the second step. So basically, so the bacteria is going to, okay, so the bacteria has receptors. So just understand the bacteria has receptors. Um, and the bacteria is going to attach to the receptors on the neutrophil membrane, right? And sometimes the good thing is that sometimes the neutrophil already mark these bacteria, right? Um, sometimes, not all the time. Not all the time it have it mark, right? Um, they can mark it like there's two types of markers. Y'all know the markers? I don't know if y'all are familiar with the markers, but there are markers. Y'all familiar with the markers that the neutrophil has to be able to spot certain bacteria?
Nope.
Like, say it again.
Nope.
Say it again.
I am not familiar, is what I'm saying.
Oh, not familiar.
Mm.
Antibodies is a type of marker, and you
Oh,
also have
that's what
complement.
you mean.
Yeah,
Okay.
yeah, yeah. Antibodies, and then chemical proteins. So
Mm-hmm.
the bacteria is going to attach to the receptors on the neutrophil membrane. That's what we mean by recognition and attachment. And the attachment is easier if the neutrophil has already marked it. Like it already have that marker and they already have that identification, right? That's the good thing about neutrophils. They remember stuff, right? But if they don't have the marker, then it's gonna be harder to remember, all right? And like I said, the markers are antibodies and complement proteins. So what's gonna happen is because it has the marker, it's gonna be easier for the neutrophil to attach to it, right? So now, like I said, the neutrophil membrane has attachments, and the attachment is going to or receptors, and they allow for like strong binding. So the first thing is attraction. Like we said, attraction is basically when the damaged tissues or what shall I say, the damaged tissues or the bacteria give off a signal saying, hey, there's a problem here. And then the neutrophils, they go there, they go to that place where there's a high concentration of either the damaged tissue or the bacteria. Then you have recognition and attachment. What happens is the bacteria is going to be attached to the receptors on the neutrophils. So the neutrophils have receptors on their membrane, and they're going to attach to the bacteria. And the attachment is easier if they are antibodies and if they are if they already have the marker like antibodies and complement proteins. The next thing we know all too well is going to be what we call endocytosis. So endocytosis. That's the second step, right? And by the way, this is not something like this is like in our notes. So if you look here, like this is basically what I'm describing. So if you look here, you see that the bacterium is attached directly to what? The neutrophil membrane. Like I said, the first step is what? Attraction, right? That's chemotaxis. That's basically the movement towards a signal. So like I said, the first step is attraction. The second step is recognition and attachment. The neutrophil has receptors, and you have the complement protein marker or you have the antibiotic marker. And I said these two markers makes it easier for like the neutrophil to attach to the bacteria. Then we have endocytosis. Can someone describe to me what they understand by endocytosis? So we have attraction, we have recognition, we have recognition and attachment, then we have endocytosis. What do you understand by endocytosis?
Endocytosis is like a process
Mhm.
where the cell engulfs substances like from the external environment
Mhm.
by forming a vesicle
Mhm.
from the cell membrane.
Very good. That's literally it. I like that you use that word engulf. So another word for engulf it's like a S word, engulf or what? SWALLOW.
Swallow.
Swallow. Yeah, that's another word. Yeah, that's another
I
word.
was
That's
like,
how
what?
Yeah, at least that's how I remember it. Like it swallows it, right? So it engulfs it. It engulfs the bacteria, right? So basically, endocytosis, let me just like make a note there. Endocytosis is basically swallowing or engulfment, right? Engulfment. That's what endocytosis is. And then, so basically, what happens is the neutrophil membrane. So if we have it right there. It's described right there, right? Right inside this nice diagram. So this is endocytosis being described here. As you can see, look at this. The neutrophil membrane, what it does, it surrounds the bacteria, right? So in endocytosis, before it swallows the bacteria, the neutrophil membrane is going to surround the bacteria first. Then it's going to have these cytoplasmic extensions, right? Pseudopodia, like they have these extensions, cytoplasmic extensions that basically form around it, right? And then it's going to take it and it's going to pull it inside. So the bacterium is pulled inside or engulfed. So that's basically the process of endocytosis. So we have attraction, we have recognition and attachment, we have endocytosis, and then we have the formation of, this number 4, the formation of phagosome. Okay, the formation of phagosome. Can someone help me with number 4, formation of phagosome? Which y'all understand by that? The formation of phagosome.
I think that's when the lysosome and the engulfed bacteria join,
Okay. Anyone else? Would
something
y'all
like that.
understand? Thank you, Charisma. Anyone else? Would y'all understand about phagosome?
I like to think of it as almost like a stomach, where
Okay.
things are going to be digested kind of thing.
So what is a phagosome? It is what and what?
It's the vesicle with the bacteria in it.
Yes, that's literally it, right? So okay, it already swallow it, right? So as you can see, it's clearly here, right? It's literally here. So as you can see, this is what we call the phagosome. The phagosome is the vesicle that's inside the bacteria. Because literally, the thing, it swallow it, right? But it's going to be inside a vesicle. And is that vesicle open or closed? Is it like an enclosed membrane or is it an open membrane? So is the phagosome the vesicle that is called the phagosome? Is it enclosed or is it open?
Enclosed.
It's enclosed. Very good. It's definitely enclosed because if you look here, it's literally right there. It's like it's a closed membrane, right? So this phagosome, so once inside the bacterium, is going to be enclosed in a membrane-bound vesicle, and that vesicle is what we call the phagosome. All right, that's literally what we're talking about there. Right. So attraction, recognition and attachment, endocytosis, formation of phagosome, and then the fifth one is going to be, let me put it there. The fifth thing is going to be fusion with lysosome. So fusion with lysosome. So it's going to start to fuse with the lysosome. Can anyone describe that process? Because something specific happened to cause it. Because remember, we have if we look at the phagosome, which is this entity right here, it is an enclosed membrane, like we talk about. So it's covered, right? So it's basically like a bubble. It's like it's inside the thing, right? So how is it going to fuse with the lysosome? Like, how is that going to happen? How's that gonna happen? Anyone have an idea? So
The
it's being described right here as well. Go ahead. I think that was Jasmine.
I was going to say through their cell membranes coming
Okay.
together.
The cell membranes coming together. Okay. Thank you. Anyone else want to try? How do we have the fusion with the lysosome?
Because the lysosome, I think it wants to kind of digest the bacteria,
Okay.
so it
Okay.
probably attaches to it, I
Okay,
guess.
good. Good. So what does okay, this is a good thing, right? Let me just put this inside there, like on the side there, maybe like in blue. All right, so let's talk about this. What do lysosomes have? So let's talk about lysosomes. What do lysosomes have? Lysosomes, they are membrane-bound organelles, but what do they contain? They contain what? Powerful, what? Digestive, y'all gotta help me now. Powerful digestive what?
Enzymes.
Enzymes, very good. What do you think these enzymes are gonna do? Remember, we have this bacteria sitting inside the the um the sitting inside the neutrophil, right? So what do you think what do you think the cell want to do with this particular bacteria? You think it just wanted to stay there and just like chill? Like what do you think it want to do with it? What does the
Digest
cell?
it.
It wants to digest it, right? Fortunately, the lysosome contain powerful digestive enzymes that could break down what? Proteins. What are the enzymes that break down proteins? Just
Protease, I think.
yeah, just add the EAS, just add that, right? And then lipids. What are the enzymes that digest lipids?
Lipase.
Yeah, yes, very good. Lipase. right? And nucleic acids, right? So lysosomes contain these powerful enzymes that could break down the proteins, that could break down lipids, that can break down nucleic acids. So the lysosome is going to move toward the phagosome, and that's what's happening here, right? Y'all could see it, right? The lysosome, you see, like, let me like make it clear to y'all. Like, as y'all could see that the lysosomes are moving toward the phagosome. Remember, the phagosome is this vesicle, right? And the lysosomes are gonna move towards it, right? Now, when the lysosome move towards the phagosome, their membranes are gonna come together, right? And when the fusion occurs, as you can see, look. So the little phagosomes are coming towards it. The lysosomes are coming towards that. The lysosomes are coming towards this. The lysosomes are coming towards this, right? And when the lysosome comes towards this particular phagosome, it's going to fuse, or it's going to come together with the phagosome. So we have the fusion or the coming together of, so let me just draw it out, right? So we have this right here is like a bunch of, let's call these our lysosomes in red, and then our phagosome is in blue, right? And what does our phagosome have inside of it? What does our phagosome have inside of it? Our phagosome have what?
Bacteria.
The bacteria. Where is the lysosome going to go? Where's the lysosome going to go? Towards what? Towards the
The
Y'all clever.
phagosome.
Very good. It's going to move towards the phagosome. So much so that it's going to fuse with it, or it's going to come together with it. Now, when you have the fusion, so let's go ahead. So we have now the fusion of, we have this. So we have the fusion of our phagosomes. We have the fusion, let me just put this here. We have the fusion now of our lysosome and our phagosome, right? So now they are one. Now, when they are one, remember, inside of these contains what? Digestive what? Enzymes. So when they become one, what also happens to the contents of these two vesicles? What happens to the contents? The contents are going to what? They're going to mix together. Y'all understand what I'm saying? The contents are going to mix together. Now, if the contents mixing together, what's going to happen to the bacteria inside? What's going to happen to the bacteria inside?
It's gonna start to break down.
It's going to break down. It's going to break down. So the bacteria has no chance because remember, notice that what does the lysosome have? The lysosome have what? Powerful digestive enzymes that's going to be able to break it down. So inside, now this new structure is called something else. I don't know if like they gave it to us. Like it's probably not important. But
Is it called a phagolysosome?
yes, it's called a phagolysosome, right? You're correct. But so inside the phagolysosome, the fusion of them, digestive enzymes are going to be released because remember, the lysosomes have the digestive enzymes. And then the internal environment of this particular phagolysosome is going to be acidic, right? So it's going to be breaking down it. So the bacterium is going to be broken down into small molecules. And then it leads us to number 6, right? This leads us to number 6. So number 6 is going to be what? What is number 6? The 6th step.
Exocytosis.
Exocytosis. Anyone else? What is the 6th step? What is the 6th step? So we have the fusion of that. Say it again.
The digestion of the bacteria.
Yeah, the digestion of it. So you could say killing and what? Digestion, right? And then after it kills it, after digest. So basically, all that is happening inside the phagolysosome. So but the bacteria is going to be killed. They are digested by proteases, nucleases, other enzymes, and it's broken down into smaller, harmless pieces, right? That's basically what happened. And then finally, it's going to be, it don't, Sierra, it don't have to be released now. Like, it could be reused by the cell, or it could be released. It depends, right? It don't have to be like, it don't automatically have to expel it. You understand what I'm saying? So, yeah, so those are the steps in phagocytosis. All right. So, can someone go over the steps? We have attraction, recognition, endocytosis. This is what we could do. We could do it like one step at a time. So who want to take attraction? Would you all remember from attraction? Anyone could take attraction? Who wants to take attraction? Okay, let me let Liam take attraction since y'all might be shy. Go ahead, Liam, take attraction. What happens in attraction step?
I was avoiding attraction to take recognition.
Say it again.
But I said I
Oh,
was
you
avoiding attraction
you
to take recognition.
okay, all right. No, I want you to take
But
attraction because you came in
no, yeah,
saying
we gonna try.
recognition. So take attraction. Go ahead. What
Okay.
happens during attraction?
So let's see.
Remember, attraction is going to be you need signals, right? Remember? Who releases chemicals?
The
Or what releases?
bacteria.
The bacteria
Well,
releases
the
chemical.
bacteria would then, right, so they would release the chemicals, which would then cause the chemotaxis. So then the neutrophils would be attracted to that and then release the antibodies and what the other thing is. I
The
forget what the other thing is.
complement proteins.
Right, correct. And
Yeah.
then that would then go into recognition.
Yeah, that definitely. So do all two of them one time then, since you're ready on it. So what he's explaining is for attraction, we have that the neutrophil is going to be attracted to the site of the infection. The chemicals are released by the bacteria or damaged tissues. And these chemicals are acting like signals. Like it's ready on the diagram, you know. Like if you look at the diagram, so this is the bacteria, right? And the first thing is that the neutrophil is attracted to the bacteria. How is it attracted to bacteria? Well, it's like a siren, right? It's like these signals, like automatically, once the neutrophil is going to, once the bacteria is inside the organism, the neutrophil is going to sense it, right? Because the bacteria gives off a chemical. Now, the bacteria is not like wanting to give off this chemical, but like the neutrophil is so designed that it could sense that the bacteria is around. And not only that, the damaged tissues also release chemicals as well that neutrophils could signal. I mean, could like they have receptors that they could be able to sense it, right? So then after they are attracted to the bacteria, then they're gonna recognize the bacteria because of the antibodies and the the complement proteins. And once they recognize it, you can finish now. I want you to talk about that.
Once they wait, where is the antibodies?
Yeah, we on that part.
So I mean, at that point, you have the antibodies as well as the complementary proteins, and at that point, we move into endocytosis because the neutrophils would now be able to recognize which, like the antibodies would almost be a beacon to the neutrophils, so they would move towards them easier and quicker. And then at that point, endocytosis would occur where the bacteria would be drawn into the cell and form its own vesicle, forming the phagosome. And then we have, well, the formation of the phagosome, and then the joining of the phagosome and the lysosomes, then the mixing of the bacteria and the digestive enzymes to then kill the bacteria, and then digestion. And then after it could be exocytosis, or to be used in the again in the future within the cell.
Very good. Very good. Sierra, you want to explain the processes as well? These 6 steps? Thank you, Liam.
What step you want me to?
You could go. He basically did all 6, so you could do the full thing as well.
Okay, so then we'll start with the first step, which is attraction. So basically, the chemicals in the bacteria is picked up by the receptors of the neutrophil.
Mhm.
So then the neutrophil will be attracted to the bacteria, the chemicals in the bacteria. And then that's when the receptors will recognize
Mhm.
the chemicals on the bacterium and become attached to these receptors, and then it will perform endocytosis,
Mhm.
where the cell membrane will engulf the bacteria,
Yes.
and then this will then form a phagosome around the bacteria
Yes.
that will contain it, and then the lysosomes, where the enzymes will become attracted to
Wait,
the phagosome.
one sec.
And
One sec. The phagosome is the vesicle. You understand?
yeah,
Not that the vesicle is what we call the phagosome, right? So that's all.
yeah.
Go ahead.
Then the lysosomes will be attracted to the phagosome and fuse
Mhm.
together with it.
Mhm.
And the enzymes of the lysosome will mix into the phagosome and start to digest the bacteria.
Yes.
And then this fusion of the lysosome and the phagosome creates the phagolysosome.
Yes.
And then by the end, the enzymes will completely either kill or digest the bacterium, or
Uh-huh.
yeah.
Yeah. So it will break it down into smaller harmless pieces, which will either be reused by the cell or expelled via exocytosis. Make sense?
I have a question.
Go ahead, Paris.
Does um the bacterium, does it have specific proteins on it that would attract the what did you say it was?
The neutrophil?
Yeah.
No. So basically, the bacteria gives off or releases signals. So it's not that it has proteins on it, but that it releases signals, chemical signals. That's why we call it chemotaxis, because chemotaxis is when a body moves towards chemical signals. So what it actually releases are chemical signals. Not only bacteria, but also damaged tissues also release these chemicals. Basically, the neutrophil is so designed that it could sense the presence of the bacteria. And so then the neutrophil is going to be drawn to that place where there's a high concentration of either damaged tissues or bacteria. All right? Hopefully that helps to clarify it a bit. Jen, Paris. Okay.
Yeah, that's it.
Right. Any other questions on the steps? Any other questions on the steps on how we do it? Okay. One more, I'm gonna let Charisma explain it, and then we can move on from this question. All right. But this is a very good diagram, and then like everything we talked about is inside the diagram. All the steps, the attraction, the recognition, the um the cytoplasmic extensions that surround the bacteria, the engulfing of it, the formation of the phagosome, right? The fusion of the lysosome and the phagosome. All these is explained in this diagram. So it is a very good diagram. And so, you should look at it carefully and, memorize it and stuff. Okay, go ahead.
Um,
Go ahead. Someone have a question?
yeah. Would we ever have to learn to draw this diagram, or would it just be the steps?
I would say learn how to draw it. I don't know if I don't know if they're gonna ask you to draw it in test one. But if you look at these questions, right? I got this from this is actually from our tutorial, but this is actually a past paper question. So it says using annotated diagrams, describe the process. So the question didn't even ask you to give it like in paragraph form. The question specifically asks for an annotated diagram. So if I were you, I would actually learn the annotated diagram, but also understand the steps that's with it as well, right?
Okay, thanks.
Yeah. Go ahead, Charisma.
I find they did really well. And explaining, do I have to?
No, go ahead. Yeah, listen. It's good when a person get because they might not understand it from me. You understand it from you. Go ahead. Say
Oh my.
charisma. Charisma say so funny. Go ahead.
Okay. Oh wow. Lord Father. Okay, so we have a bacterium or bacteria
Hmm.
that releases chemical signals,
Yes.
right? So therefore, the neutrophil, which is the first responders to infection,
Yes.
are attracted to it.
Hmm.
And because the bacteria releases the receptors and right, so it is attracted to it,
Hmm.
and therefore the neutrophil engulf the bacteria
Yes.
and it form a phagosome,
Yes.
and the lysosome fuses with the phagosome to form a phagolysosome,
Hmm.
where the lysosome starts to break down the bacterium,
Huh?
and therefore it kills and digests it and release it through a process called exocytosis.
Yes. Yes. Very good. Very good. Very good. All right.
Does the
So
phagosome also release chemicals?
no. So basically, good question. So if you see the structure right there, Paris, this structure is the vesicle containing the phagosome. I'm sorry, containing the bacteria. This blue structure right there, that is what we call the phagosome. It is a combination of the vesicle along with the embedded bacteria. That's literally the phagosome. These red structures that we have here, that's the lysosomes. They contain the powerful digestive enzymes such as the lipases, the proteases, the nucleases that's going to digest proteins, lipids, and nucleic acids. These are going to be drawn to the phagosome, right? And eventually they're going to fuse with the phagosome to form the um, lysosome, that word, right? But basically it's going to form a structure that is a combination of the lysosome and the phagosome. And then the contents that's inside the lysosome, which is the digestive enzymes, they're going to be disperse into the cytoplasm such that it's going to be able to digest these bacteria and reduce it into harmless molecules. And these harmless molecules are going to either be they're going to either be excreted or disposed of via exocytosis, or sometimes they can be even reused in the cell. So that's what's happening here. But the phagosome itself does not contain any chemicals for digestion, only the lysosome. That's where
So then how does the lysosome become attracted to the phagosome?
um I don't know. I don't know the process of like what causes the attraction. I just know that it is drawn towards it. But that's a good question. I
All
don't know
right.
how it, yeah. Okay, so good. So we're gonna do here is I'm gonna let y'all do a little quiz based on this. I'm gonna send the link inside the chat and then um and then we can do it and then we're gonna move on to the rest of the questions, right? So let me put the link in the chat. It's also gonna be a link and it's gonna be a QR code and you can choose which way you want to do it. It's basically a Kahoot. I don't know if you all are familiar with Kahoot, but Kahoot is really nice. I like using Kahoot. And so we're gonna do it. We're gonna um so give me one moment. Let me just put the link inside the chat and then we can do it together. It's a good way just to revise the things that we have talked about.
Isaac, can I request a class?
When?
Anytime before the exam.
Another one,
Yeah, but just on the heart,
just on the
like
heart?
heart and the blood and stuff.
Okay. We could do that. We just have to, I was thinking about that and I was like, we should have another day because
Yeah, because they're bad like that. All right, bye now.
oh no, 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 that's not what I meant. I meant because we need it. Shoot,
No, I mean like you're good. You're good at it. You're good at teaching.
we need it. Um, so I was thinking about that because we wouldn't have enough time to cover everything. I was thinking about it. We should do it. We should definitely do it. I don't know when people are gonna be free, but we should do another one just before it's time to take it, just for a good revision on the heart. If not, I probably just I don't know. We'll see. I'll probably just put it inside the chat. Okay, so let me start it and send y'all the quiz link. Okay, there's the quiz. Just log on to that. I'm also gonna send the QR code as well. So just join with your name. So it's basically like thank you, Sierra. It's basically like a group thing that we're gonna do together. Sierra, you never came for your stuff, or we never met.
When can I come and pick it up? I don't know.
I don't know. We should meet in class, but I don't know. I don't know. Whenever you see me in class, then just let me know.
Okay, no problem.
Okay, we have Angelina, Charisma, Liam, Paris, Adrian. It's 6 of us.
I'm coming.
Okay. Okay, so we just have to wait on Jasmine. And anyone else who wanna join, you could join. Demia, you wanna join? And Shekinah, you wanna join? And okay, Angelina is here already. Okay. All right, so okay, Shekinah, good. We have everyone. So basically, let me just tell you how this works. This thing works based on speed. So the question is going to be placed on the screen. Only the question is going to be seen on my screen, right? The question and the options. On your screen, all you would see would be like, you would see the colors, red, green, yellow, whatever. You have to click on the correct color that corresponds with my screen. So your screen would not have the question. Only on my screen would have the question. All right? And just make sure that you do it fast, because if you do it slow, then you're not really gonna get as much points as you would get if you were to do it fast. Okay? And whoever wins, whenever you see me, I have something for you. Okay, so let's begin. Where's Jasmine? Jasmine get knocked out again. Jasmine, what happened? You
I left the page for a second. I'm coming back.
okay.
Oh Lord. It's trying to reconnect, but it's not going through.
Oh, see, you have Wi-Fi issues.
Maybe.
There you are. There you are.
Okay.
All right, let's go. All right, so first question. Question number 1 states, which cell performs phagocytosis? Which cell performs phagocytosis? Is it the erythrocyte, neutrophil, neutron, or platelet? Which cell performs phagocytosis? Is it the erythrocyte? Very good, it's the neutrophil. Very good, guys. Next. What forms after engulfment? What forms after engulfment? What form after engulfment? Is it phagosome, peroxisome, ribosome, or endosome? What forms after engulfment? Very good. It's going to be the phagosome. Very good, guys. Next question. Which enhances bacterial attachment? Or what enhances bacterial attachment? Collagen, melanin, ATP, or antibodies? What enhances bacterial attachment? Collagen, melanin, ATP, or antibodies? What enhances bacterial attachment? Very good. Antibodies. Remember you said that antibodies are like the markers and they make it easier for the attachment to take place. All right, next. Very good, guys. Chemotaxis guides cells via gradients, vacuoles, hormones, or myosin. Chemotaxis guides cells via gradients, vacuoles, hormones, or myosin. It's definitely gradients. You all say hormones. Okay, let me explain this. So yes, a signal is produced, but the neutrophil is going to go to the place, remember I said it? The place of higher concentration. What's another word for concentration? That's basically like a diffusion gradient. So it's going to go where the gradient or that concentration is higher. All right? So the correct answer is gradient. All right, everyone stayed the same. Okay. Next, which structure fuses with the phagosome? Which structure fuses with the phagosome? Lysosome, Golgi, mitochondria, or nucleus? Which structure fuses with the phagosome? Lysosome. Very good, the lysosome. Next. What kills inside the phagolysosome? What kills inside phagolysosome? Keratin, RNA, hemoglobin, or ROS? Yes, ROS. I don't think I told you all that, but that's it.
What is
What I
our
supposed RS is basically like, okay, let me just show you all it's in the next slide. I don't have it in front of me, but it's in the next slide that I supposed to show you all the next slide before I gave you all the quiz. All right? But yeah. Next. Which movement wraps bacteria? Yeah. Pseudopodia, flagella, villi, or cilia? Pseudopodia. Those are gonna be the cytoplasmic extensions. Very good. Okay. Okay. Next. Where do attachment chemicals originate? The bones, the hair, the liver, or the bacteria? Very good, bacteria. They release the chemicals. Very good. Next. What terms means coating microbes? What term? Oh, I didn't give you all this. Let me skip it. But don't answer this. I didn't give you all this. But the correct answer is this one, the O1. I mean, y'all could still put it, but opsonization, that means coating microbes. Yeah. All right, next. All right, so we have two more. What is formed after fusion? What is formed after fusion? Phagolysome, proteasome, exosome, or lysate? Yes, phagolysome. Very good, guys. Two more questions. Which receptors bind bacteria? Which receptors bind bacteria? This one is easy. Which receptors? These receptors are gonna bind to the bacteria. Ion channels, micromembrane receptors, ribosomes, or centrioles. Definitely the membrane receptors, the neutrophil membrane receptors. Very good. Next, which neutrophil process internalizes? Which neutrophil process internalizes? Mitocytosis, endocytosis, transcription, or exocytosis? Which neutrophil? Very good. Endocytosis. Good stuff, guys. Let's see how y'all did. Very good. Who's that is? Sierra. Very good, Sierra. Very good, Liam. And who's this one? Charisma. Very good. And who is fourth and fifth? Hi, y'all. Jasmine, Paris. All right. Y'all did pretty well. And everyone else. Y'all did pretty well, man. So good. Good stuff. So that's that. Just a little thing for us to just to go over and stuff like that. All right, so let's move on. Let's finish up with this, right? So, but good job, y'all. Y'all did pretty well on that. Okay, so this is the slide that I was supposed to show y'all. Wait, let me go back before I gave y'all the quiz, but you know, I just gave y'all the quiz without it. But this slide explains what we did. So this is the diagram, obviously, and then this slide is explaining everything we talked about: the chemotaxis, the recognition and attachment, the engulfing, the fusion and digestion, all them stuff, right? And the ROS is just basically going to be the reactive oxygen species. That's going to be inside the neutrophils. It's produced by the neutrophils to help them kill pathogens. I don't think we need to know that, but that's just something that I found that is helpful. All right. Okay. So good. Let's move on to the next question. So the next question in our sheet is, let's go back. Do you all have tutorial 3 open? Let me go to the next question on tutorial 3. It says so the first question was the annotated diagram. The next question was let me just put it there. To list the differences between neutrophils and between lymphocytes. So can someone tell me any difference that you know between neutrophils and lymphocytes? What are some differences that y'all know?
A neutrophil has a multi-lobe nucleus, whereas lymphocytes only have a singular circular nucleus.
Say it again.
Neutrophils have multi-lobe nucleus, whereas lymphocytes have one circular nucleus.
Yes, that's correct. I can accept that. Anyone else?
The neutrophils are non-specific while the lymphocytes are specific.
Okay, yes.
The neutrophil, they contain fine granules in the cytoplasm, and the lymphocytes, they don't have much cytoplasm, so they have little amounts, and sometimes you would see granules, and sometimes, most cases, not.
Yeah, very good. That's correct. I agree with that. Anyone else? So definitely the shape of it. Neutrophils are multi-lobed. They have a segmented nucleus. Lymphocytes, they are large. They have a round nucleus. Let me just give you all the table one time so that we wouldn't take too long. So this is the table. The nucleus for the neutrophil. I think who said that? Sierra. Yeah. Multi-lobe nucleus. Lymphocytes, large round nucleus. Neutrophils, the position. The nucleus occupies a less space. This is one of the a good identifying mark. The neutrophil is not going to cover the whole entire cell. But the lymphocytes, the nucleus occupies most of the cell. That's going to be very important when it's time to look at the diagrams and see what is what, all right? The cytoplasm amount, moderate amount of cytoplasm, very thin rim of cytoplasm. Why do you think that the lymphocytes only have a thin rim of cytoplasm? Why do you think that is the case? Anyone can answer. Why do you think it only have like a thin rim of cytoplasm?
What was that? The lymphocyte?
Yeah, why does you think the lymphocytes only have a thin rim of cytoplasm? Goes right back to the second point. This.
That's to push out the um. I'm
No, it's right.
thinking of it.
It's right there. It's literally right there in the second point.
I know
Right
what
there.
should I trying to think it logically in my own head.
Oh, you trying to? Okay, no problem. Go ahead.
But yeah, so that the nucleus occupies most of the cell. But I was thinking like, you know how because they're part of the cell mediated response, because they're in specific immunity, I was thinking like, you know, in the plant, right? How the vacuole takes up most of the space
It does. It
in
does.
the upper epidermis to push the chlorophyll out so it could take the sunlight. I was thinking about it like
Oh,
the
okay.
antibodies. It's not the antibodies, but basically the receptors on the outside of the lymphocytes would be pushed out as well.
Okay.
Or does that not have anything to do with it?
I'm not sure, but you could look into it. I never even thought about that, but it's a good parallel. Yeah. But double check it though.
Okay.
But it definitely have I just was thinking like physically, like why would it have only a thin rim of cytoplasm? Because the nucleus is gonna occupy most of it. That's all. Just very
Okay.
simply. Yeah. Cytoplasmic granules. Like I think Chris was saying, the neutrophil is gonna have granular, fine, visible, um, granules, but the lymphocytes, it's not gonna have visible granules all the time, right? So no visible granules, and we're talking about in a light microscope, right? So you could basically say that neutrophils, they have fine granules, but then lymphocytes in general, they don't have visible granules, all right? The cell size, the neutrophil is larger and the lymphocytes are smaller. So here's a diagram showing both of them. On which side do we have the neutrophil? Like which side, the left one or the right side? The neutrophil now.
Left.
The left, right? I know we have the label there, right? But if you were just to look at it, right? How could you tell the difference between it, like just looking at it? How would you know?
The lobe nucleus or nuclei, and then the granules as well as the amount of cytoplasm.
Very good. So we would say that, where are we? Paris, how would you be able to tell the difference between these two? I know we have the labels, but how would you be able to tell the difference between these two cells?
I mean, you would have relied on the nucleus.
Say it again?
I mean, you would have relied on the nucleus.
Yeah. So this one is lobed, multi-lobed, and this one is one large nucleus there. So it's pretty clear. And then these ones have, you could see the granules, like you were saying, and this one, no visible granules. So neutrophil, lymphocyte. Okay, so pretty
Oh,
clear
can I
stuff.
just say?
Yeah, go ahead.
Yeah, by the way, when I did biology the first time, we did have to look at a picture of the cells. Of course, it was in black and white and terrible quality, but you did have to label which one was the neutrophil and which one was the lymphocytes.
Really?
Yeah.
Wow, look at that. All right, so it's good to know them, right? Thank you for that. All right, so the next question, it says, describe how cell-mediated immune response occurs in the presence of a pathogen, for example, a bacteria. Your account should include the following under separate headings: origin and maturation of specific cells, and steps in the mediated and steps in the cell-mediated response. All right. So we want to describe how the cell-mediated immune response occurs in the presence of a pathogen, for example, bacteria. Your account should include origin and maturation of specific cells and steps in the cell-mediated process. So Let's begin with the first one, right? So we have to talk about its origin. And when I say origin, we're talking about where it come from, like where it's located, where it start from, all right? And maturation, obviously talking about its progression, how it matures, how it develops. And we're talking specifically about these specific cells. Then after we talk about where it come from, its origin, we need to talk about the steps in the response that it gives to when an invader or bacteria comes in. So this actually asking us for two things. Number one, we want the origin and development. And number two, we want to know how does this cell respond when a pathogen comes and invade in a specific environment, right? So obviously this has 4 marks, so we're going to try to at least aim at 4 things. And then here we have 2 marks. So we're going to aim at like, not I say 2 marks, this has 6 marks. So we're going to try to aim at 6 things, all right? So let's begin with the first one, origin and maturation of specific cells. Anyone can answer, tell me how we're going to proceed. What y'all thinking? So we're going
You'd
to,
have to compare the B cells and T cells, right?
yes, go ahead.
Listen, I just remember B cells is from bone marrow.
Okay, all right, no problem. Well, that's helpful. That's helpful. That's the origin. That's where it come from. So let's talk about that, right? So we're gonna say that the T lymphocyte. Anyway, before we continue, anyone else wanna chime in?
The B lymphocyte, they produce antibodies. And the T lymphocyte, they are in, I think, immune response. I'm
Okay.
trying to remember. They destroy like cancers or infected cells, and
Okay.
they regulate immune response.
Okay. Anyone else wanna chime in? Adrian, you wanna chime in? Alicia, Jasmine?
The lymphocytes originate from the thymus. Well, they
Okay.
mature. They
Yeah,
mature in the thymus.
yes, they do mature on the thymus. That's correct. So we have that. We have their origin. Their origin is obviously in the bone marrow, right? And we have their maturation place in the thymus. Very good. Anything else? So let's talk about that, right? I'm specifically going to talk about the T lymphocytes. So let's talk about that. So the T lymphocytes, let me just quickly have the T lymphocytes, they're going to originate. Where we say they originate again? T lymphocytes. Where? From where? Which location?
The bone marrow?
The bone marrow. Very good. From stem cells in the bone marrow, right? You know, bone marrow, like for persons here who's eat meat, anyone here is vegetarian? Is there anyone here who's vegetarian? Oh wow, y'all missing out. Y'all missing out. But anyway, for those persons who like eat the chicken bone, you know, like when you're eating the chicken bone and then you like bite it, and then you see those like weird stuff inside of it, that's the bone marrow, right? Some people actually swallow that. Y'all swallow that? I don't know. Some people actually some people actually swallow that. But
I
that's the
know my
bone marrow.
dad does.
Yeah, some people really just do that, right? They really love it. So I mean, that's their thing. So, but that's the bone marrow right there. That's where we have the origination of the T lymphocyte. So they're gonna originate in the stem cells in the bone marrow, and they're going to mature. So after they originate in the bone marrow, they're gonna mature where? Where do we say that they're gonna mature? We said the specific place that they are gonna originate from the bone marrow, but where are they gonna mature?
The thymus gland?
The thymus, very good. So immature T cells migrate. So these immature T cells, they're gonna migrate to a place where they could develop. So they migrate to the thymus. What else could we say? The thymus. It's a gland, right? So let's say gland, the thymus gland, right? So immature T cells migrate to the thymus gland. What else could we say? We could also talk about what happens in the thymus gland. So in the thymus, we could talk about how it differentiates in the thymus, right? So how they mature and So we know that they migrated to the thymus, but we got to talk about how the fact that they do mature in there, right? So they, in the thymus, they, speaking of our T cells, they mature and what? Differentiate into specific, let's just say specific T cells. For example, I don't know if you're familiar with the helper T cells and the cytotoxic T cells. Let's say cytotoxic T cells. So we talk about where they originate from. That was our first statement. We talk about where they mature and they develop. I think we're pretty much done. But the only thing is this is for 4 points, so we need to add something else. Just something like a filler. So we know where they originate, we know where they mature. We said that the immature, they migrate to the thymus. Do the T cells have receptors? Do they have receptors?
Yes, they do.
Yes, they do. So we got to talk about that. Let's just mention something about that. Let's just say that each T cell develops, still talking about their development, right? Develops specific receptors. What these receptors are going to do? What's the function of the receptors? They're going to do what?
They're going to recognize specific
yeah.
antigens.
Yeah, let's say that recognize particular or recognizes, let's say, receptors recognize a particular antigen. Good. And that's our 4 points, right? So that's that. So now, so let's summarize it. They, you know what? Let me let Adrian. Where's Adrian? Where's Adrian? Adrian, are you there? Oh, probably shy.
Yeah.
Okay, you wanna summarize our 4 points first, and in your own words, maybe?
What was that? Say that one.
Like if you want to summarize the 4 points that we have for part I, we're basically talking about the origin and the maturation of the specific cells.
T cells, well, the T lymphocytes, they basically come from the bone marrow.
Yeah.
And they stem from the stem cells, which, like, I guess in babies, they would use stem cells to regrow or help them develop, basically. So
yeah.
immature T cells migrate to the thymus gland.
That's correct. Yeah.
And within that thymus, they mature to different specialization. So the T cells, T helper cells, and the cytotoxic T cells. So it's developed into 3 different types of cells within that maturation. So they would either go either or route, basically. And the T cells further go on to the receptors to recognize a particular antigen or like the antigen to respond to the antibodies.
Very good. So, like you're saying, we talk about where they originate from in the bone marrow. We talk about how the immature ones migrate to the thymus. We talk about how they differentiate into different ones. And we talk about how each one of them are going to grow specific receptors to be able to identify the antigen. Okay? Cool. So that is the, what do you call it? Origin and maturation. So can I move this? Let's talk about the steps in the mediated process. Okay. And by the way, like an antigen is just basically like an ID or a tag on a pathogen. Okay. Okay, cool. So let's move on to, so I'm gonna move on to this. So I'm gonna move this and we're gonna talk about part number 6, the steps in the mediated process. So cool. So it's 6 steps. So we want to mention like some certain things. So we want to talk about the steps. So let's talk about steps in the cell-mediated response. Okay. So we're going to try to at least mention 6 things, right? So Sierra, set us off. How are we looking? We're going to do well. Where's Sierra? Oh, so I can't. Oh, Sierra's not here. So let's go to maybe Alicia. Alicia. Or Jasmine. What are some steps in the mediated process? Or in the immediate response?
I'm not sure for this one.
You're not sure about this one? Okay. Liam, you wanna try?
Immediate response on how what?
So we know phagocytosis. Phagocytosis is the engulfing, right? But this one is talking about like you know, in a more specific level, like what happens. We have immune responses for obviously phagocytosis, but then we have immune response for on a more specific level that's more precise, right?
Right.
What happens
So
in that?
what
Yeah.
I remember is so you have B lymphocytes and T lymphocytes, right? I forget how they split up though.
Okay.
But what I do remember is that there's presence of antibodies and antigens, which help in the breakdown. Because what happens is I think one of the T cells basically breaks down one of the parasites or whatever, and then when it starts to replicate itself, it produces that is it antigen or antibody on its surface?
Okay.
Something
yeah,
like
you on the right track. I like that you mentioned keywords, antigens, antibodies, pathogens, those stuff, right? So when we talk about, I'm not gonna write this whole thing down because it's like too long. I'm just gonna like summarize it, right? So basically, an antigen is basically like I said, it's basically an ID tag, and we know ID tags at UWE, where we have ID tags. ID tag is just basically something for recognition, right? So, good. The immune system is going to recognize what, like, invaders are not, like, belonging to the immune system. So it's going to recognize the tag and say, hey, you're not supposed to be there, right? So where can we find these tags? Well, you could find the tags, or you could find the antigens. So like I said, it's the tag that is on the pathogen. So we have this pathogen, right? So this is a pathogen. So we have the pathogen here. This is the stuff that the body don't like, right? And on this pathogen, you're gonna have a tag. So this is the tag. The tag is like the ID, right? That's gonna be your antigen. And that ID is gonna tell the immune system, hey, that particular pathogen does not belong here. And it's going to react against it. Now, where's this pathogen? Where's the ID found? Well, the ID is going to be found on the surface of the bacteria, right? It's going to be found on toxins. It's going to be found on foreign cells. Like transplant. You ever heard of like transplant? When someone get a transplant. So if someone get a transplant, let's just say you get a transplant from, like you say, you transplant a kidney, someone donate the kidney or whatever, right? Do you know that the body performs an immune response on the foreign kidney? You all know that? I don't know if you all are familiar with that. But if you get a kidney from someone else, your body is going to consider that as foreign, and your body's going to react against it. That's why persons who get transplants, they get something called immunoresponse, like not immunoresponse, immunosuppressants, right? Which basically is going to suppress their immune system such that their immune system is not going to attack that particular organ, right? So the antigens are found on pathogens. The antigen is found on viral particles. The antigen is found on toxins. The antigen is also found on like organs that are not supposed to be there, like transplanted organs, right? But that's antigens. Now, so the antigen is going to be detected. Who detects the antigens? Well, the immune cells, they basically gonna recognize it. They're gonna recognize they're gonna recognize these antigens, and they're gonna recognize them using specific receptors. We talked about the receptors earlier when we talk about the origin and maturation of specific cells. So you're gonna have receptors, and the receptors are going to recognize the receptors on these white blood cells. They're gonna recognize the antigens, right? Like the ID tag. Now B cells, they produce antibodies that bind specifically to the antigen, and the T cells, they're gonna be activated to destroy it. Okay, and so that's basically the idea. Okay, in summary. Now, let me go here. This is like the full thing. If you want like the full everything, all the 6 points, you could take a screenshot of this and like get like it step by step. But Paris, can you read it just so we could have like a full step by step thing? I just wanted to explain it in general, and then you can always take a screenshot of like the specific of it.
Okay.
Yeah.
So a pathogen infects body cells, and antigens from the pathogen are displayed on the cell's surface. An antigen-presenting cell, example, macrophage, presents the antigen to a helper T cell. The helper T cell becomes activated and releases cytokines. Cytokines stimulate the activation and clonal expansion of cytotic T cells specific to that antigen. Cytotic T cells bind to the infected cells, displaying the antigen. Cytotic T cells release perforin and enzymes that destroy the infected cell.
Very good.
That's all.
Yeah. All right, cool. All right, so we're done with that. So you can take a screenshot of this. I also will be sending this document inside the chat. So, you can have it. Okay, this is the previous slide. Someone want me to go back to the previous slide. So this is the stuff on maturation that we talked about. And then this one is the stuff on the immune response, okay? So since our time is dwindling, I want us to go to, we have other things that we can look at, but I want us to look at, we did all this. I want us to look at this. So cool. So this is, obviously I'm gonna send the whole document. So any question that we didn't do, obviously I have the answers is gonna be in the document. So you can look at it, but I want us to go over this, right? So let's look at this diagram, right? So on the vertical or y-axis, we have pressure. And on the horizontal x-axis, we have region of the blood system. So here, what sort of activity is going to be described at A? What is happening? Can someone describe to me the pressure in region A? How would you say? Relative to like other portions.
It's high pressure.
High pressure, very good. So, Adrian, describe to me the general trend of this diagram. Starting from A to E. What's the general trend? Adrian.
So I would say due to the partial pressures by A, it had like a downward trend
Yeah, it's a downward trend. So
until
it's,
so
yeah.
the antigen, oh gosh, the volume of the blood.
Well, we're just talking about the pressure at this point on the side. So the pressure starts high and it continuously goes what? Like you were saying, like a downward trend, right?
Yeah, downward, like the partial pressure of it continuously goes down.
Yeah, so the pressure continuously goes down, right? So we start at a high pressure and it continuously go down and down and down and down, right? Now this is talking about blood pressure changes as blood flows through various portions of the blood system. What region is A? What region is A?
Highest concentration of blood.
Yeah, not only you, Adrian, anyone else. What region is A? What region is B?
It should
What
be the
region is C?
artery.
What region is D and what is E? Say it again. What is A?
I think that it should be the arteries
A should be the arteries. Very good. Why is A the artery?
because the arteries, they will have like the highest pressure.
Very good. Very good response. A is the artery because the artery is going to be under highest pressure, right? So after the arteries, then you have B. Who does the artery give the blood to? So we have arteries, then what? We have what?
The arterioles.
Very good. The
Veins.
arterioles. So we have arteries. So the region A is the arteries. Region B is the arterioles, right? It doesn't have high pressure, but it still have some pressure, right? Certainly more pressure than C and D and E, right? So we have the arteries, then we have the arterioles, right? Then what would be region C? So we have arteries, we have arterioles. What is region C? And even lower pressure, but what is region C?
Is it venules?
Say it again.
The venules, I think that's how you pronounce.
The venules. That's region D. Right? That's region D.
Um,
Region D is the venules. So we
so
have
so
arteries. We have arteries for A. We have B is going to be the arterioles. D is the venules, and E is the vein. What is C?
The capillaries.
The capillaries. Yeah, C is the capillaries, right? So based on that,
Wait, what are venules?
venules are like the version of, like arteries have arterioles, veins have venules. So think of venules as they have the same function as veins, but they're smaller, right? They're just smaller. So veins are gonna be a little bit more bigger, but venules are gonna be smaller because they can be sending the blood to the crevices, the more smaller areas of the body, right? So artery is big. Arterioles perform the same function of arteries, with just smaller arteries. Same thing with veins. Veins, big blood vessels. Venules perform the same function as veins, they just are smaller. So basically a smaller version of the veins, basically, right? So good. Does that make sense, Paris?
Yeah, and you said the venules was E or the
No.
veins?
Arteries is A. The B is arterioles. C is capillaries. D is D is veins, and E is
Okay.
venules. So, okay, let me just label it, right? So let's label it. So this is a question that they gave us. So we have I, double I, triple I, and IV. Which one is the correct answer? A, well, sorry, it's ready there. All right, complete giveaway. A is the artery, double I is the arteriole, triple I is the capillary, and IV is the venule. So there you have it in live color, right? So this is another way that they can ask this question. Okay, next, let's answer the question. We never answered the question, right? So the question is, let's go here. Calculate the drop in pressure from the start of region B to the end of region D. Can someone walk me through how we're gonna do this question? We're gonna calculate the drop in pressure from the start of region B to the end of region D. How are we gonna do that? Show working. So Let's go ahead and do that. Anyone have any answer? Liam, you want to help us? What we doing now?
Which thing? Which region?
Start of region B to the end of region E.
Okay, so A is B, right?
So yeah, start of region B, from the
Mm-hmm.
start of region B to the end of region E.
Okay, so B would be arterial, C would be capillaries, D would be venules to go back to the veins, and then E would be veins.
Yeah. So we looking at, so let me just let's write it down. So this is the artery.
Mm-hmm.
Yeah. This is the arterioles. This is not clear at all. Let me just erase it and make it clearer. So obviously you have different regions and different regions have different things. So this one, let's go ahead and, so what is region A? Guys, what is region A? Region A is the, y'all help me.
Arteries.
The artery is very good. Region B is the,
Arterioles.
very good, arterioles. It's hard to really write so small, but I'm gonna try. All right. Yeesh, yeesh. Yeah, okay. Region C is the what?
Capillaries.
Capillaries, very good. Capillaries. Everybody's spelling it wrong, but y'all get the picture.
Mm.
All right, and then region D is the,
Venules.
no, and
Oh,
the
no,
venules,
not. Wait,
the
veins.
veins first, yes. Then region E is the
Venules.
venules, very good. Good. Now, this question is asking us what area, what calculate, so we're gonna have to do some maths here, right? Calculate the drop in blood pressure from the start of region B to the end of region D. So what is the, so that means we're gonna do it like this. We're gonna say, hey, what is this line? Can someone tell me what is this going to be? So this is the end. This is E. This is the end of region E. What is this in terms of our pressure? We're going from 0 to 5. What is this one line going to be?
0.5.
Who said that?
Me.
Elisha. Very good. That is 0.5. You're smart.
I counted it.
Very good. Very good. All right. So what is going to be the beginning of region B?
That should be
So this is 15.
15.5.
Yeah, 15.5. 15.5. The rest of y'all, do y'all see it? This is 0.5. This is 15.5. Y'all see that? Let me know if y'all see it. So we have 0.5 and we have 15.5. Right? So cool. So we want to calculate the drop of it. What's going to be our answer?
The answer should be 15
Very good.
kPa.
Very good. The answer is going to be 15 kPa. Right? So what we're going to do is subtract 15.5 with 0.5. kPa is going to give us 15 kPa. Right? Good. Does this make sense to y'all? Let me know.
Mm-hmm.
Liam, we good? Adrian, Jasmine?
Right.
Good. Good. Very good. Okay, cool. All right, so next, let me move from this. Move from the slide. Let's move on to, so we got the drop. And by the way, these are the identifications that I talked about. The arterioles, the arteries, arterioles, capillaries, venules, and veins. We already like label it already, so that's good. All right, let's move on to the next question. The next question says, Explain what brings about the drop in pressure between B and D. So what brings about the drop in pressure between B and D? What is B and D? B is what? B is arterioles, right? And D is venules. So what brings about the drop in pressure between the arterioles and the venules?
I'm not even sure if I'm gonna be saying this right, but
Say it
like,
again. Go ahead. Say it again.
I'm not sure if this is like the right terminology, but the fact that the blood has to go around the body, and therefore the pressure would continuously drop.
Okay, yeah, friction. Yeah. So that's true. Um,
The size of the vessels.
yeah, the size of it. Because think about it, right? So remember, if we're looking at an artery is big compared to so this is an artery, right? So you have artery. You have artery, right? So this is like your bigger pipe, right? And this is like a lot, right? Then you go to the arterioles. The arterioles are so small. They're like this, right? They're very small. Not only small, but they're gonna you're talking about the arterioles, but then you're also gonna talk about the capillaries as well. So the arterioles, they have an even more narrower lumen, right? So if it's more narrower, that's like bad English. But anyway, if it's more narrower, what's gonna happen? You're gonna have an increased resistance. Would you think? Because think about it. If you are like a molecule or whatever, if you're a molecule and you walk in through air, you pass in freely. But if you pass in through air, obviously it's gonna be what? Much harder to pass through air, right? So it's gonna have a greater resistance because of the smaller lumens. So it's gonna have a high level of resistance to blood flow, and that's gonna cause friction, and the friction is gonna reduce the blood pressure, right? And then also too, by the time you get to the capillaries don't play. The capillaries are a network. The capillaries are a network of blood vessels, right? So what does that mean? That means it's gonna go here. That means it's gonna go here. That means it's gonna go here. It's gonna be like a whole network of things, and they connected and stuff like that. And the capillary is like a whole network of stuff, right? So let me ask you a question. If you have, let's say for example, right? Let's say for example, you have an option. You have an option for one person to blast you with but now it's the same amount of water now. Same amount of water. That's all I remember that. So you have an option for one person to take do y'all have like water balloon guns over here? Like, so if you have like a water balloon gun, not a water balloon gun, but like a gun that you can put water in and it spray people. Do y'all have them stuff over here? Like basically like you just press it and like it squirts out the water. But think about like a machine. Y'all have stuff like that?
Yeah.
Okay, yeah. So think about a machine like that. Would you rather one person to spray you with all the water they have, so think about the artery, or would you rather 10 persons to spray you with smaller guns? That's the thing now. So you have one person with the big gun and the same amount of water, and you have 10 persons with much, much, much more smaller guns spraying you. What would give you the most pressure? Or what would give you the most force? What do you think? The one person with the big gun? Exactly, the big gun. Because the persons with the small gun, it's gonna be so small because their gun's so small, plus it's the same amount of liquid. So by the time they squirt at you, you ain't gonna really feel it because it's dispersed. That's the same thing with the capillaries. The capillaries, it is a network, and so it increases the cross-sectional area. And so what's going to happen is it's going to reduce the pressure because it's a network, and it's like a lot of them, and blood is going to flow through here, blood is going to flow through here, blood is going to flow through. So by the time all that happened, the pressure is already reduced, right? That's why pressure from the arteries, by the time it get to the arterioles, obviously the narrow lumen resistance reduces pressure. Then you have the capillaries, the network of capillaries that is going to have a huge cross-sectional area, which is going to lower the pressure again, right? And so energy is going to be lost, or the force is going to be lost because of the resistance and because of the network of the capillaries. So by the time it reaches the venules, it's lost most of the pressure. All right. And also the distance too, the distance it have to travel, because blood passing through the arteries is pretty much fresh from coming from the blood, from coming from the heart. So it's gonna have a higher pressure. But by the time I get to the arterioles and then the capillaries and the vein and like the distance is going to cause like the pressure not to be so high. You all understand what I'm saying? So like that's why it has a drop in pressure, right? So hopefully that helps a bit with that. Okay, so next. So this next slide explains everything we'll be talking about. Like I said, I'm gonna send you this document, so you could take a screenshot of this, or you could just wait until I send a document, or just get a good understanding of it too. And we already talk about all these. We talk about the resistance. We talk about the friction and all that stuff. Okay, so suggests why it is important that pressure in region C is not as great in pressure in region A. Why is it important for pressure in region C in the capillaries to not be as great as the pressure in region A? Why is that important? Why is it important for the pressure in the capillaries to not be as great as the pressure in the arteries? Can someone, Liam, you wanna try? Why is that important? Or maybe
And C being the capillaries, right?
yeah, C is the capillaries and A is the arteries. So why is it
The
important?
arteries.
Yeah, why is it important for the pressure in the capillaries to not be as great as the pressure in the arteries?
The capillaries have much smaller single cell walls, much thinner single cell walls than the arteries, which are triple layer with thick what's it
Muscular
called?
walls, muscular walls.
Thick muscular, but I'm thinking of the the middle layer.
Oh, you mean that particular
Media.
term?
Yeah.
Yeah.
Was it media intima?
I don't remember.
I don't remember. But
I
yeah.
don't remember either. But it does have more elastic fibers. So it could tolerate the scratching. It could tolerate it could tolerate that, right? Because of the muscular walls and because of its scratching ability because of a lot of elastic fibers. But the capillaries is only one cell thick. What are you trying to do? If it has a lot of pressure coming to the capillaries, what do you think is gonna happen
Yeah,
to the capillaries?
it would rupture.
It would rupture. That's the word. It's gonna rupture. So the important is that you don't want to have a lot of pressure in the capillaries because it's gonna rupture the capillaries. All right, so that's that. Let me see what someone say in the chat. Could you go, Bob? Okay, so cool. It would rupture. Very good. Yeah, Damia said the same thing. Cool. So that's what the same thing is saying. We said that it's important because it's one cell thick. It will cause them to rupture or burst. Okay, so that's that. Let's move on to this one. So it says, state which of the regions A to E shown on the diagram represents the capillaries. Anyone could answer this. State which of the regions A to E shown on the diagram represents the capillaries. What region that was? Was that A? Was that B? Was that C? Or was that D? Or was that E? Which region represented the capillaries? Y'all remember? Which region represented the capillaries?
It should be C.
It would C. It would C. Yeah, it would C. All right, next. So the capillaries represented by region C. Good. All right, so next, select two structural features of capillaries and explain how each feature helps with the exchange of materials between blood and the tissue fluid. So we have the feature and we have the role in exchange. So the capillaries are designed in such a way that they could be able to help with the exchange of materials between the blood and the tissue fluid. That's the way how the capillaries are so designed, right? Or so structured. But what are structural features that helps the capillaries to perform that function, whereby it facilitates the exchange of materials, right? So we have feature and we have role and exchange. Go ahead. Someone want to say something?
I can
see.
go.
Go ahead.
Oh, okay.
So Alicia, then sorry, Jasmine, then Alicia.
Okay. So it's thin walls or 1 cell thick, which allows for easy diffusion.
Yeah. 100%. Anyone else? So it's 1 cell thick, so it provides basically a short diffusion distance, so it allows it to rapid diffusion of oxygen, carbon dioxide, and stuff across it. Anyone else? What's another feature of the capillaries that allows for it to perform its function? Alicia is gonna say something.
You say again?
Go ahead, Liam.
What did she say?
She said the 1 cell thick. The cell is the wall is 1 cell thick.
Oh, that allows for easy diffusion of gases and nutrients from the blood into the muscle tissues.
Yes, that's exactly right. So that because it's 1 cell thick, it allows for easy diffusion. What's another feature? Because they say 2. We talk about the 1 cell thick. What's another feature?
I had to think about this for a little bit because, well, she said exactly what I was going to say.
Yeah, fine, no problem. Charisma, you have something? You could help us?
But
go ahead,
I thought something else.
Go ahead.
I was thinking about no, don't say sorry. Be proud. Be proud that you're very smart.
Go ahead. Go ahead.
I was thinking about how extensive capillaries are. Like there's just a lot of them.
Yes.
And they're able to go to almost every single tissue, right?
Yes. So it provides a surface area,
Mm-hmm.
right? It provides a large surface area for diffusion. That's the point right there. So the extensive branching network that you talked about, it provides a large surface area for diffusion. So it allows for more diffusion to take place because of the greater surface area, right? So that's a good point. Very good point. So,
Thank you.
So we have 2. We have the wall is 1 cell thick. We have the large surface area because of extensive branching, and that allows for greater diffusion. Anyone else want to chime in with something else before we move on? They only asked for 2, so you could give more, but they only asked for 2. Anyone else? Good. Okay, so there are more, but we're not gonna like spend our whole entire life on that. Cool. So let's go. So this is like I gave you all extra. Like obviously, you could find other more. We have 1 cell thick, narrow lumen, branching, whatever, stuff like that. Cool. This is what I want to look at. So it says substances are exchanged between red blood cells, plasma, and tissue fluid. Complete the table to show which of the statements apply to each of these components of the body. So we have the fill in each box using a tick to show that the statements apply or cross if it doesn't. So we have the statement, plasma, tissue fluid, cytoplasm of red blood cells. We have contains hemoglobin, contains water, contain antibodies in direct contact with muscle cells. So we have plasma, we have tissue fluids, and we have cytoplasm of red blood cells. So we're going to write a tick to show that the statements apply or X if it doesn't. So let's start with the statement which is contains hemoglobin, and we're talking about plasma, tissue fluid, cytoplasm. So Does plasma contain hemoglobin? Do we gonna put a tick or we gonna put an X? Does plasma contain hemoglobin?
I think we should put X.
Who's speaking?
Alicia.
Alicia, yeah, you're definitely right. It's an X. Plasma does not contain hemoglobin. Does plasma contain water?
Yes.
Yes, plasma does contain water. Does plasma contain antibodies? Charisma. Where's charisma? Charisma, are you there?
I think so.
You think so? Jasmine, you think so?
You know, I'm not gamble. I think so.
You think so? Okay. All right, you all are correct. It does contain antibodies. Yeah. Is it in direct contact with muscle cells?
No.
No. No, so definitely not. Then tissue fluid. Does tissue fluid contain hemoglobin?
No.
No, it doesn't. That's inside the cell. Does it contain water?
Yep.
Yes, it has to have water. Does it contain antibodies? It's inside the cell now. Yeah, it can have antibodies. Is it in direct
yeah.
is it in direct contact with muscle cells?
Yeah, that's what I was hesitating.
Yes.
I was like, eh.
Is in direct this tissue fluid.
Yeah.
So it's in direct contact with muscle cells, right? Now, cytoplasm of red blood cells. Does it contain hemoglobin?
Obviously.
Yeah, 100%. They was waiting for that one. And then, does it contain water?
Yep.
Yes. Does it contain antibodies? Red blood cells. Do red blood cells contain antibodies?
No.
No, it doesn't, right? No, red blood cells, their main function is gas, to carry oxygen. So no.
Yeah.
Is it in direct contact with muscle cells? The cytoplasm of red blood cells is in direct contact with
No.
No, it's not. All right. So this is the table completed. That makes sense? Good. Next, let's move on. So we have the table completed, and this is it. Okay, now it's time to label these things. So we have cell B, we have cell C, we have cell A. What is B? Let's start with C. C, I think C is the easiest one. What is C?
Yeah, those are red blood cells.
They're the red blood cells. What do we call the red blood cells? What's a fancy word that we use?
Erythrocytes.
Erythrocytes, very good. Yeah, so C is the erythrocytes. Then we have, I think A is the second easiest one. What is A?
Lymphocytes.
A is the lymphocytes. What gives it away?
The lack of cytoplasm in a large nucleus.
Very good. It has only a thin layer of cytoplasm. You see, like I know it's hard to see, right? So let me just like recreate it. So basically, what this is trying to show is you have a cell, and you have on the inside you have the nucleus, which takes up like the majority of it. So this dark portion, this dark region here, is really the nucleus, the extra dark part. And then you see the lighter part. The lighter part is like, it's supposed to be the cytoplasm, right?
Yeah, if you squint, you can see it. Basically.
Yeah, if you squint, you could see the difference between the shades, right? So the darker part is obviously the nucleus. It has a large nucleus, and then the smaller region has the cytoplasm. So this is definitely a lymphocyte. Now, B, what is B? B is what?
Neutrophil.
Neutrophil, very good. What gives it away?
You can see the multi-lobe
Very
nucleus.
good. The multi-lobe nucleus. So if you look carefully here, We have this guy here, this guy here.
You can see like 4 segments. Yeah.
Yeah, it's a multi-lobe nucleus like that. So you could see it. So cool. So this is the, so let's go ahead and label it, right? So this is the lymphocyte. A is the lymphocyte, B is the neutrophil, and C is the erythrocyte. Make sense? Good.
Mhm.
Good. Next, we have, so we have it listed right there. Okay, so this I just did like a diagram I saw just to compare it, so you could see the difference and whatnot. And then I also have another diagram here, just so you could see it. As you can see for the lymphocyte, it has like a big nucleus. Neutrophil has multi-lobe nucleus, and you can see the big difference here, right? Moving right along, let's move on to question number II. It says outline 2 features of the cell C that enable it to carry its function. So cell C is the erythrocyte. What are some features of the erythrocyte or the red blood cells that help it to carry out its function?
It is it has a concave shape.
Close.
Yeah,
Bicon. Very good. Biconcave shape. Yes. And what does that do? That increases the surface what?
Surface area.
Very good. Increases surface area and helps with diffusion, right? So that's a good one. Biconcave shape. Increases the surface area. What else it has?
A lack of a nucleus.
A lack of nucleus. How does that help?
It helps more oxygen to bind.
Yes, so it basically creates more space, right? It provides more space for hemoglobin. And so that means that the red blood cells, if it has more hemoglobin, then it has more oxygen carrying capacity, right? It could be able to carry more oxygen because of more hemoglobin, right? Then, well, we need to mention it, right? It has hemoglobin, right? So hemoglobin is a good thing. So we have 3 so far. The biconcave shape, right? The fact that it has no nucleus, and the fact that it has hemoglobin. It does it have any organelle? It lacks most organelles, right? And it has a flexible membrane. So all those things help it to carry out its function. Okay. The main thing I would mention here, I would mention the biconcave disc shape, and I would mention the fact that it has no nucleus, if I were only mentioning 2. Okay. Moving right along, we have, so this is like, you could look at it, you could see the different functions here. Okay, list 2 visible differences between nucleus, well, cell A and cell B. We already talked about it, multi-lobe nucleus, large nucleus. And then the next one is that, like basically the difference between neutrophil and lymphocytes, we would already like outlined the same picture that we talked about a long time. I mean, originally, one has very little cytoplasm, one has more visible cytoplasm, stuff like that. Okay, next we have give one function for cell B. Cell B is a neutrophil. What does neutrophil mainly help with? We start off this session with that.
Neutrophil is the first responder to infection.
Okay, what main process does it help with?
What process does it help with?
Yes.
This or would you say humoral response?
Say it again?
Would you say phagocytosis?
Yes, phagocytosis. Yeah,
Could
that's
you
literally
not, if you say humoral response with that, what do you think?
Say it again if you say.
If you were to say the humoral response.
I'm not understanding what you're saying. Type in the chat.
Humoral response, he said.
Humoral response? What is that?
Some kind of response.
Hold on. Let me see if I'm retarded or not.
Humoral. Spell that. I never heard a humoral response.
Humoral.
Humoral response. What is a humoral response?
Oh wait, no, that's
adaptive immunity. It's saying here, Morse.
Wait, hold on.
This adapted immunity. Let me see. Neutrophil.
Because it has
Yeah,
cell-mediated
it does. Yeah, it says it acts on human. Yeah, you could. Yeah, it's right there, so it could work. I mean, that's not the
Yeah.
first thing that comes to my mind. The first thing that comes to my
Yeah.
mind is phagocytosis off the top because that's literally like the main one. We just
Yeah,
talk
I
about
just
go ahead.
okay.
Go ahead.
I was just thinking of, if there could be other possibilities.
Oh yeah, of course, of course.
You know
That's
what I mean?
definitely one. Yeah,
Yeah.
definitely one. Humoral response. Yeah, cool. So that's one. Obviously, phagocytosis is like the main one. And the other ones, like you say, engulf and destroy bacteria. First line of defense, trigger, release antimicrobials, sorry, antimicrobial substances. And like Liam says, it has a role in humoral immune response by destroying by destroying pathogens. Basically talking about phagocytosis anyway. Anything else on this thing? We have this question. And that's like our last question, but it's like 8:30. So we're gonna end here officially. So I'm gonna end the recording. And if you all have any questions, I'm gonna end the recording because I don't want the recording to be too long. But if you have any other questions, you can let me know. All right? So let me end the recording.